Let's now discuss male reproductive system. The major male reproductive functions are secretion of sex hormones, then production of sperm, and transport of that sperm from male to female. And the reproductive organ is testis. Now basically the testis has the loops of convoluted seminiferous tubules. These seminiferous tubules, in the walls of these seminiferous tubules are the germ cells, primitive germ cells, and which undergo the process of spermatogenesis, formation of spermatozoa. Seminiferous tubules would drain into the epididymis to vas deferens, and then in this route there would be ejaculation of sperm. There are between the nests of cells there is interstitial cell of Leydig. These are the interstitial cells which synthesize the testosterone. And the third component of the testis is Sertoli cells. So let's look at these components one by one. The seminiferous tubules and the process of spermatogenesis. Now basically the fluid in the lumen of the seminiferous tubules, its composition is very different from plasma, in the sense that this fluid has very less glucose or proteins, and it is rich in androgens, estrogens, potassium, inositol, glutamic acid, aspartic acid. So that's the fluid in the lumen of the seminiferous tubules. Formation of sperm takes place in the seminiferous tubules, and the spermatogenesis has basically three distinct phases. Let's try to understand these three distinct phases. First stage is the stem cell renewal and the production of spermatogonia. Second stage is germ cell proliferation, and the third stage is spermiogenesis. So the first phase, as I said, stem cell renewal and production of spermatogonia. The primitive germ cells in the walls of seminiferous tubules, they will undergo mitosis. They will divide by mitosis to form the primary spermatocytes. That will be the first phase. Second phase, this primary spermatocytes will undergo meiotic division, and this will reduce the number of chromosomes, of course. It's a two-stage process. They will divide into secondary spermatocytes and spermatids, which will contain the haploid number of 23 chromosomes. And the third phase is spermatids will form or mature into the spermatozoa. Now that process is called, final stage is referred to as spermiogenesis. So these are the three phases or stages during spermatogenesis, and mitosis and meiosis both they require the cyclins and cyclin dependent kinases. They are the essential mediators of mitosis and meiosis. Now, a single primitive germ cell, or each spermatogonium rather, will give rise to 64 spermatozoa, and the process of spermatogenesis takes about 74 to 75 days. Well, the sperm production, the most reliable expression of sperm production is called as the sperm production rate, and it is the daily number of sperm cells produced per gram of testicular parenchyma. So a 20-year-old man would have the production rate of about 6.5 million sperm per gram of testes per day. That's said to be the most reliable expression of sperm production, or called a sperm production rate. Now, spermatogenesis itself differs from the oogenesis in the female in various ways, but three most important aspects in which spermatogenesis differs from the oogenesis. In the female, the mitotic proliferation of the oogonia, or the germ cells, takes place entirely before birth. Whereas in the male, the spermatogonia proliferate only after the puberty, and then it continues to divide. This process continues throughout life. So that's the first. So we have already seen that the females have to contend with a decreasing number of ova, whereas in the case of male reproductive physiology, there is a regular production of spermatozoa throughout life. Second important difference is the meiotic divisions of the primary oocyte produce only a single matured ovum, whereas in the male, the meiotic divisions of the primary spermatocyte would produce 4 spermatozoa, 4 mature spermatozoa. So that's another difference. And the third important difference is, in the female, second meiotic division is completed after the fertilization, immediately after the fertilization, and after that there is no further processing, there is no further development of the cell would take place. Whereas in the male, the products of meiosis, that is spermatids, they undergo substantial further differentiation to produce mature spermatozoa. So that's another difference between the two. Looking at the sperm, it's an intricate motile cell rich in DNA and with a cap which is known as acrosome. Now, apart from the other things, with the sperms, this cap would contain an enzyme called as hyaluronidase, and this is necessary for the digestion of hyaluronic acid so that the sperm can penetrate the cervical mucus. Then it also contains a special form of angiotensin converting enzyme, which is called as germinal angiotensin converting enzyme. The function of this is not known, but the male mice which are deficient in this enzyme are have known to have reduced fertility. So another less known factor, but well related with the fertility. Now, so far as the gametogenesis is concerned in males, what all things are needed? The factors that regulate the spermatogenesis are 2 cell types, the Leydig cell and the Sertoli cell. We will see the functions of Sertoli cells separately, but the Leydig cell will produce testosterone, which concentration of which should increase locally for the maturing sperm. Sertoli cells, they nurture the developing spermatozoa. They have a nursing function to perform. 2 gonadotropins, LH and FSH. LH, which is also called as ICSH, interstitial cell stimulating hormone, because this acts on the interstitial cell of Leydig to form testosterone, which is necessary for the spermatogenesis. And FSH, another factor necessary for the spermatogenesis. So FSH and testosterone, two important factors necessary for or in factors that influence this process of spermatogenesis. Pituitary gonadotropins LH, FSH, the testosterone by the Leydig cell, and the two cells, Leydig cells and Sertoli cells. These are the factors involved in the process of spermatogenesis. Now, the mature sperm acquires motility in the epididymis, but not immediately. After staying for 48 hours in the epididymis, the sperm acquires motility. It's called as progressive motility, and a protein which has been found to be involved in acquiring motility is cat spur. This is said to be a calcium channel involved in the calcium current to develop through it, and it's said to be an important unique protein in the spermatozoa involved in acquiring motility for the sperm. What are the functions of Sertoli cells? Sertoli cell, it forms the blood testis barrier. It's in that there are tight junctions between the adjacent Sertoli cells which form the blood testicular barrier. Sertoli cells secrete a hormone called as inhibin. So here we have the Leydig cell which synthesizes testosterone, and Sertoli cell which synthesizes inhibin. What is the difference between these two hormones? Testosterone will exert the negative feedback on the pituitary gonadotropin LH as well as on the hypothalamic GnRH. Inhibin has only one negative feedback, inhibition of FSH. So that's its primary role. Inhibin inhibits the FSH secretion from the pituitary. Apart from this, what are the other functions of Sertoli cells? They nurture the spermatozoa, nursing function for the developing sperm. Sertoli cells synthesize Mullerian inhibiting substance or Mullerian inhibiting factor for the regression of the Mullerian duct. So, some of the important functions of the Sertoli cell.